Welcome to Computational Biology. Today, we are embarking on a fascinating journey into the very blueprint of human life, the human genome. Imagine trying to read a book that holds the secrets to our health, our traits, and our evolution. But the book is written in a microscopic chemical language. In this lecture, we will explore how computer science and biology join forces to decode this massive puzzle. We will learn how we sequence the DNA, the mathematics behind ensuring we read it correctly, and why this is the ultimate foundation for modern personalized medicine. Let us dive in. To truly understand the challenge of sequencing the human genome, imagine you have a massive, ancient encyclopedia. This book contains all the secrets of human life. But there is a catch. It has been shredded into millions of tiny pieces. Your job is to tape it back together to read the story. You cannot just read it from start to finish. Instead, you have to look at overlapping snippets to figure out how the whole puzzle fits together. This is exactly the challenge scientists faced. It is a massive computational puzzle where the pieces are microscopic chemical letters, and solving it requires the perfect marriage of biology and computer science. Now that we have an intuitive grasp, let us formally define what we are working with. The human genome is the complete set of nucleic acid sequences for humans. It is encoded as DNA within the 23 chromosome pairs in our cells. In computational biology, we do not just look at this as biology. We treat this genome as a massive text string. And when I say massive, I mean approximately 3.2 billion base pairs long. These base pairs are represented by the chemical letters A, C, G, and T. Reading a string of 3.2 billion letters is a monumental task that pushes the limits of modern technology. Because this text string is so incredibly long, we face a major technological hurdle. Current technology simply cannot read this entire string at once, from end to end. To solve this, we use a clever technique called shotgun sequencing. This method intentionally breaks the genome into short, readable fragments that a sequencing machine can easily process. Once we have these millions of short fragments, computational algorithms take over. They align and merge these fragments based on their overlapping sequences to reconstruct the original, complete genome. It is exactly like finding the overlapping words in our shredded encyclopedia to rebuild the pages. Understanding how we sequence the genome is fascinating, but why does it matter so much? The human genome is the ultimate foundation of modern personalized medicine. By knowing the standard sequence of human DNA, we can easily identify tiny mutations that cause severe genetic diseases, like cystic fibrosis or cancer. Furthermore, it allows researchers to perform genome-wide association studies. These studies help find the specific genes that make someone susceptible to complex conditions, like diabetes or heart disease. Without a reference human genome, we would be completely blind to the genetic root causes of human health. It literally serves as the biological blueprint guiding every major medical discovery today. This brings us to a critical mathematical question. How do we know if we have generated enough short fragments to cover the entire 3.2 billion letters? We cannot just guess. Computational biologists use the Lander-Waterman equation to calculate sequencing coverage. The equation is C equals N times L divided by G. Coverage simply means the average number of times any single letter in the genome has been read. In this formula, C represents the coverage. N is the total number of short reads generated by the machine. L is the average length of each short read, and G represents the total length of the target genome. If we increase the number of reads, or the read length, our coverage increases, giving us more overlapping data and a much more accurate assembly. Now that we have our formula, let us see this with a concrete example. Suppose a sequencing machine generates 60 million short reads to sequence a small bacterial genome. Each read has an average length of 150 base pairs, and the total genome size is 3 million base pairs. What is the coverage? First, we identify our variables. N is 60 million, L is 150, and G is 3 million. We plug these into the Lander-Waterman equation. Multiplying the number of reads by the read length gives us 9 billion total DNA letters successfully read. Dividing this massive number by the genome size of 3 million gives us our final answer of 3,000. Physically, this means every single letter was read on average 3,000 times, ensuring highly reliable data. To truly cement this idea, let us look at a visual schematic of sequencing coverage. At the top of this diagram, you see a solid horizontal line representing the entire target genome, labeled G. Below it are many smaller staggered lines. These represent the millions of short reads generated by our sequencing machines, each of length L. Notice the vertical dashed box highlighting a specific section of the genome. If you count the number of short reads that fall within that specific vertical slice, you will see exactly 4 overlapping lines. This visually demonstrates a coverage of 4x for that specific base. By generating enough of these overlapping short reads, we ensure that the entire target genome is covered multiple times, allowing us to accurately reconstruct the full sequence without leaving massive gaps. While calculating average coverage is incredibly useful, it leads us to an important realization. Average coverage does not directly tell us if we missed any spots. Because the DNA fragments are generated randomly by the sequencing machine, the distribution of reads is not perfectly even. By pure chance, some parts of the genome might be read 50 times, while other parts are completely skipped. These skipped parts become blind spots in our data. To truly understand the quality of our sequencing experiment, we can't just rely on the average. We must mathematically calculate the probability of these unsequenced gaps occurring. To understand our blind spots, we use a special probability formula derived from the Poisson distribution. This formula calculates the exact chance of missing a letter. The equation is P equals e to the power of -c. Here, P represents the probability that any specific base in the genome is completely missed, meaning it has 0 coverage. The symbol E is Euler's number, a mathematical constant approximately equal to 2.718, which is heavily used in probability. Finally, C is the average coverage we calculated using the Lander-Waterman equation. This elegant formula tells us that as the coverage C increases, the probability P of missing a section drops exponentially toward 0. This perfectly explains why high coverage is absolutely critical. Let us apply this probability formula to a real-world scenario involving the human genome. Suppose a researcher is sequencing the human genome, which has a massive size of 3.2 billion base pairs. They generate just enough reads to achieve an average coverage of 5x. What is the probability that a specific base is missed? First, we identify our coverage, C, which is 5. We plug this value into the exponent of Euler's number in our Poisson formula, giving us P equals e to the power of -5. Calculating this exponential decay function yields approximately 0.0067. Physically, this translates to a 0.67% chance of missing any specific base. It seems small, but in a massive genome, this fraction is highly significant. Now that we have the probability of missing a single base, we must answer the second part of the question. Roughly how many total base pairs of the human genome will remain completely unsequenced? To find this, we take our resulting probability of 0.0067 and multiply it by the total size of the human genome, which is 3.2 billion. The mathematical result is 21,440,000 base pairs. Physically, this means that despite reading the genome 5 times over on average, over 21 million letters of the human genetic code are still missing. This massive gap highlights exactly why modern human genome sequencing requires much higher coverage, typically around 30x, to be clinically useful for patients. We have covered a tremendous amount of ground today. Let us review the key takeaways. First, we learned that the human genome is a massive 3.2 billion letter sequence. And because we cannot read it all at once, we rely on shotgun sequencing to piece together overlapping fragments. Second, we explored the Lander-Waterman equation, which allows us to calculate our sequencing coverage to ensure we have enough data. Third, we discovered that due to the randomness of sequencing, we must use the Poisson probability formula to calculate the exact chance of leaving unsequenced gaps. Finally, we saw mathematically why high coverage is absolutely essential to eliminate blind spots, making modern genetic medicine and personalized treatments possible. You now understand the computational foundation of the human genome.